Assalamualaikum warahmatullahi wabarakatuh. The Honorable the Second Sriwijaya International Conference on Basic and Science Committee. My name is Dina Pitasari. I will present my team research article entitled In Vitro Anthelmintic Activity of Sapanwood, Jasalpinia salpan lignomethanol extract against ringworm Ascaridia galli. Introductions. Infection with the nematode worm Ascaridia galli is one of the most common gastrointestinal nematode in poultry worldwide, result in weight loss. Severe infection with Ascaridia galli can show symptoms of dropiness, bleeding, and diarrhea. Treatment is needed to overcome health problem caused by worm infection, especially Ascaridia galli. Things to consider are efficacy, toxicity, drug resistance, and drug residues left in food. It is necessary to develop the use of deworming drug derived from plant as an alternative to the use of synthetic drugs. Sapanwood has several benefits, including antibacterial, antifungal, antioxidant, and antiviral. The result of previous studies showed that the internal extract of sapanwood contains flavonoids, saponins, alkaloids, and tannins. According to Hamza et al. 2016, this compound provides anthelmintic activity against Ascaridia galli. However, from the literature research, there is no research related to the anthelmintic activity of sapanwood. Therefore, the study aims to determine the anthelmintic activity of sapanwood extract in vitro. Material and methods. The study used several materials such as sapanwood, 96% ethanol, aqueous, 0.9% sodium chloride, some reagent, sodium CMC, mebendazole tablets, and the test animal were Ascaridia galli. Sample processing. Sapanwood was obtained in Bengkayang, West Kalimantan, Indonesia. The determination was carried out at LIPI, Bogor, Indonesia. Measuration was carried out using 96% ethanol as a solvent by the chemical screening test for the presence of alkaloid, flavonoid, saponins, and tannins. In vitro assays. Adult Ascaridia galli collected from small intestine free-range chicken obtained from Sungai Ambawang Kubudaya Regency, West Kalimantan, Indonesia, which were freshly slaughtered. Collected worms were washed with sodium chloride each time to remove dirt. Worms are selected that are still actively moving. Worm size 7 until 11 cm, no anatomically deformed. The sample size determination was calculated using Federal formula so that 5 worms were obtained per treatment groups. Sapanwood ethanol extract was used at a concentration of 2.5, 5 and 7.5 mg per milliliters. Mebendazole as a positive control with a concentration of 10 mg per milliliters. Sodium chloride 0.9% as negative control. The treatment group was incubated into the control temperature 37 degrees Celsius for 24 hours. Worm mortality was observed at 1 hour interval for 24 hours. Observations were made on worms that experienced paralysis and death. Percent inhibition of worm mortality applied. To evaluate the anthelmintic activity in vitro, the LC50 value was determined. From the observation data obtained, observed mortality was calculated. The data were processed using Microsoft Excel. Result and discussion. Sample preparations. The water content of Simplicia was obtained at 6.43% with an extract yield of 4.52%. Ethanol is selected based on the solubility of the secondary metabolite compound from the sample. The color of extract was brownish caused by the Brazilian pigment of sapanwood in ethanol solvent, which produced an orange to brownish red color. Chemical screening of sapanwood extract was described in Table 1. The result indicate that the extract sapanwood contains alkaloid, flavonoids, saponins, and tannins. In vitro assay results. Sapanwood contain bioactive compound, one of which is thought to be ability against Ascaridia galli. In this study, the anthelmintic activity of ethanol extract of sapanwood was tested, which consisted of several concentrations including 2.5, 5 and 7.5 mg per milliliters, positive control using mebendazole 10 mg per milliliters, and negative control using 0.9% sodium chloride in vitro. In this study, the motility Ascaridia galli worm were given ethanol extract on sapanwood with observation interval every 1 hour for 24 hours after incubation. Based on the data on average number of deaths of Ascaridia galli, it is suspected that mebendazole onset data at the 3rd hour is concentration of sapanwood ethanol extract at 5th and 4th and 3rd hour. The mebendazole half-life data based on the study result were at 7 hours, its concentration of sapanwood ethanol extract at 8th, 7th, and 7th hour. The data of average percentage of mortality for each test group can be seen in Table 2. From the data, it can be concluded that the 3 concentration of ethanol extract of sapanwood and positive control have anthelmintic activity with a percent mortality of 100%. The following is relationship between concentration log and probability values on worm mortality. Based on these equations, the calculation of LC50 24-hour using Microsoft Excel is known that the LC50 24-hour of 7-weight ethanol extract against Ascaridia galli is 0.37 mg per milliliter. The enzymatic activity was caused by the presence of alkaloids, flavonoids, saponins, and tannin in the saponwood extract based on the results of its phytochemical screening. The enzymatic activity of alkaloid compound is on nervous system and also act on the human digestive tract. Flavonoid antioxidants play a role in enzymatic activity through modulating bioactivity. Flavonoid can inhibit the secretion of several enzymes such as tyrosine, cholinesterase, esterase that cause nerve muscle paralysis by disrupting neural and muscle function and ultimately causing the death of worms. In addition, flavonoids can affect the nervous system of worms, causing the nerve degenerations, resulting in disruption of nerve impulses that cause the worm to become paralyzed and die. Saponin contained in saponwood extract can affect the physiological stability of worms. In addition, saponin can integrate into cellular membranes, resulting in damage to membrane permeability and disrupting ion homeostasis between intra and extracellular. This compound in plant or plant extract can also provide cytotoxic activity. Tannin compounds have enzymatic activity through three types of mechanisms: the connection of tannin to free proteins, enzyme inhibition, and membrane damage paralytic effect cause the digestive metabolic process to be disrupted, reduces the availability of nutrients, and ultimately result in the death of larvae. In addition, tannin also have officidal activity by depositing protein that make up the outer layer of the egg, causing cell division to not occur in the egg so that larva formation does not occur, or connecting to the larva cuticle, where it is rich in glycoproteins, causing the death of worms. Conclusions. The finding of this study expected to contribute the development of medical plant research, especially saponwood. The results show that the ethanol extract of saponwood had enzymatic activity against roundworm Ascaridia galli in vitro with percent mortality of the 3 sample concentration of 100% and the LC50 24-hour value of 0.37 mg per milliliter. The recommendation for further research is to test the enzymatic activity on the other worm species and develop the bioenzymatic product from ethanol extract of saponwood. We thank to the research team of Academy Farmasi RC Pontianak for their contributions. I will close my presentation with a quote: "Let's create the change we wish to see in the world with research." Thank you very much. My name is Dina Yuspitasari. Wassalamualaikum warahmatullahi wabarakatuh.